Hello, all dear students. I am Mrs. Kiran Yogesh Bhokre, working as an assistant professor in Department of Chemistry at Sinhagad College of Pharmacy, Vadgaon, Pune. Hereby going to teach you a topic which comes under the biochemistry urea cycle. So, in this today's presentation, I am going to teach about urea cycle, energetics of urea cycle, significance of urea cycle, and regulation of urea cycle. So, we'll in general discuss about the first urea. So, urea is the end product of the protein metabolism, or you can say amino acid metabolism, and the nitrogen of amino acids converted into ammonia is toxic to the body. So, it is first converted to the urea and then detoxified. The urea cycle is the first metabolic pathway to be elucidated. The cycle is known as a Krebs-Henselt urea cycle. Ornithine is the first member of the reaction, so it is also called as a ornithine cycle. Then urea is synthesized in liver and transported to the kidneys for the excretion in urine. Then the two nitrogen atoms of urea are derived from two different sources, one from the ammonia and the other directly from amino group of aspartic acid. Then carbon atom is supplied by CO2. This you will going to understand with reference to the structure of urea. Then urea is the end product of protein metabolism, or you can say amino acid metabolism. Urea accounts for 80 to 90% of the nitrogen containing substances excreted in the urine. Urea synthesis is a 5-step cyclic process with 5 distinct enzymes. The first 2 enzymes are present in mitochondria while the rest are localized in the cytosol. So this is nothing but overall the urea cycle in general representation, where you can see first step is the formation of carbonyl phosphate, then second formation of citronil, then third formation of arginosuccinate, fourth formation of arginine, and then finally ornithine, which is regenerated in the cycle, and they will going to form the urea. So this is in general about the representation of the urea cycle. Then we'll see one by one in detail the steps about the urea synthesis. First of all, the first step is the formation of carbonyl phosphate. So by the condensation of CO2 and NH4 with the utilization of 2 ATP going to form a compound carbonyl phosphate. So second step is the formation of citronil. This ornithine is react with the form carbonyl phosphate to form the citrulline. Then third step, formation of arginosuccinate, where citronil reacts with the aspartate with the utilization of 1 ATP to give the compound arginosuccinate. Then fourth, formation of arginine. The arginosuccinate form, it is cleaved by the enzyme arginosuccinase to give a compound arginine and fumarate. And the formation of urea, finally fifth step, is formed by the reaction of arginine with water in presence of the enzyme arginine to give urea and ornithine. So these steps are overview for the synthesis of urea. So in detail we'll learn from all the steps by the urea cycle representation. So this is nothing but the urea cycle, where you can see that this left side part, the reaction which takes place inside the mitochondria, while as the right side part where the reaction generally takes place in the cytosol. So we'll see one by one. So the first step, as we had seen, the condensation of CO2 plus ammonia takes place to form a compound carbonyl phosphate, and this reaction takes place by the catalyzation, or you can say this catalyzation reaction is generally carried out by the enzyme carbonyl phosphate synthase, which is also known as a CPS1. So now this is the first step, that is formation of your compound carbonyl phosphate. Then in the second step, this carbonyl phosphate reacts with the ornithine in presence of the compound or you can say this reaction is catalyzed by the enzyme ornithine transcarbamylase to form the compound citronelle. The citronelle is nothing but the combination of your carbamyl phosphate plus the combination of L-ornithine, and this citronelle, there is representation of the citronelle in this form of this structure, where you can say there occurs the addition or you can say generation of first NH2 group of the structure of urea. Okay, so then again this citronelle when reacts with the aspartate in the next step, that is the third step, and the reaction is catalyzed by the enzyme arginosuccinate synthetase enzyme, whereas there occurs from the L-aspartate compound the generation of second NH2 group inside the structure of formation of urea, and this reaction also consumes 1 ATP to form a compound arginosuccinate. Okay, so there occurs a formation of compound arginosuccinate in the third step, form L-citronelle plus L-aspartate. So here there is a formation of a compound arginosuccinate. Then this arginosuccinate compound is again reacts, or you can say the reaction is catalyzed by the enzyme arginosuccinate to give formation of two compounds fumarate plus L-arginine. And as we know, the L-arginine is formed as a precursor for the synthesis of your compound urea. So this L-arginine reacts with, or you can say the reaction is catalyzed by the enzyme arginase to form the compound urea, and this reaction required the 1 mole of water to give the compound urea. So this is nothing but the 5-step urea cycle. Then we'll see one by one all the steps in detail involved in the urea cycle synthesis or formation of urea. So first step, as we had seen, the formation of carbamyl phosphate. Carbamyl phosphate synthesis CPS1 of the mitochondria catalyzes the condensation of NH4 plus ions with the CO2 to form carbamyl phosphate. This step consumes 2 ATP and this reaction is irreversible, and this is also a rate limiting step. Then secondly, CPS1 requires N-acetyl glutamate for its activity, which is also known as a NAC. Then carbamyl phosphate synthesis 2 CPS2 involved in pyrimidine synthesis, and it is present in cytosol. It accepts amino group from glutamate and does not require N-acetyl glutamate for its activity. This is about the carbamyl phosphate synthesis enzyme second. Then first step, the formation of carbamyl phosphate. Here you can see, as we had seen in the presentation, CO2 plus NH3, which utilizes 2 ATP in presence of the enzyme carbamyl phosphate, synthesizes first to give the compound carbamyl phosphate. With this reaction also requires an acetyl glutamate. Then second step, this is regarding the carbamyl phosphate synthesis enzymes CPS1 and CPS2. So CPS1, it is present in mitochondria, whereas CPS2 in cytosol. CPS1 utilizes NH3, whereas the CPS2 utilizes glutamine. CPS1 is required, or you can say required for the synthesis of urea cycle, whereas CPS2 is used in the pyrimidine biosynthesis. Then CPS1 is activated by NAC, as we had seen, whereas the CPS2 is inhibited by CTP enzyme. Okay, so the second step, formation of citronelle. The second reaction is also mitochondrial reaction. Citronelle is synthesized from carbamyl phosphate and ornithine by ornithine transcarbamylase. Ornithine is regenerated and it is used in the urea cycle. So second step, formation of citronelle. Ornithine, when reacts with carbamyl phosphate in presence of ornithine transcarbamylase to give citronelle. Then third step, formation of arginosuccinate. Citronil condenses with the aspartate to form arginosuccinate by the enzyme arginosuccinate synthetase. Then second amino group of urea is incorporated in this step. It requires ATP, it is cleaved to AMP and PPI. Two high energy bonds are required and immediately broken down to inorganic phosphate. So fourth step, formation of arginine or cleavage of arginosuccinate. The enzyme arginosuccinate or arginosuccinate lyase cleaves arginosuccinate to arginine and fumarate, an intermediate in the TCA cycle. Fumarate provides connecting link with the TCA cycle or gluconeogenesis. Then the last step, formation of urea. Arginase is the fifth and final enzyme that cleaves arginine to yield urea and ornithine. Ornithine is regenerated, enters mitochondria for its use in the urea cycle. Arginine is activated by CO2 plus and Mn2 plus. Ornithine and lysine compete with arginine, that is a competitive inhibition. So arginase is mostly found in the liver, while the rest of the enzymes of urea cycle are also present in the other tissues. Arginine synthesis may occur to varying degrees in many tissues, but only the liver can ultimately produce urea. Then energetics of urea cycle. The overall reaction may be summarized for the urea cycle is NH3 plus CO2 plus aspartate, which gives rise urea plus fumarate. Two ATPs are used in the first reaction. Another ATP is converted to AMP plus PPI in the third step, which is equivalent to 2 ATPs. The urea cycle consumes 4 high energy phosphate bonds. The fumarate formed in the fourth step may be converted to malate. Then significance of urea cycle. First is toxic ammonia is converted to intoxic urea. Then synthesis of semi-essential amino acid arginine takes place. Ornithine is the precursor for proline. Then polyamines synthesis, and polyamines include putrescine, spermidine and spermine. Then polyamines have diverse roles in cell growth and proliferation. Then regulation of urea cycle. Carbamoyl phosphate synthase CPS1 is the rate limiting enzyme in the urea cycle, and CPS1 is allosterically activated by N-acetylglutamine, or you can say NAC. It is synthesized from glutamate and acetyl-CoA by synthase and degraded by a hydrolase. Then the rate of urea synthesis in liver is correlated with the concentration of N-acetylglutamate. Then this is how the formation of NAC, or you can say N-acetylglutamate, takes place. So N-acetylglutamate is synthesized from the glutamate and it is hydrolyzed by the enzyme NAC hydrolase with generation of acetate molecule. So this is nothing but the reaction, how, or you can say representation of a structure, how the formation of NAC will takes place and its degradation. Then summary. In this presentation, you are learn about urea cycle, energetics of urea cycle, then significance of urea cycle and regulation of urea cycle. So from this presentation you can summarize or you can understand about the urea cycle, energetics of urea cycle, significance of urea cycle and regulation of urea cycle. Thank you.